where we mostly start the process of extracting the DNA and preparing for the PCR and indexing PCR, for example, when we want to do amplicon sequencing of the 16S region of the microbes. So we process here all kind of different samples.
So my name is Emily, and I study at the Technical University of Munich, and I'm doing my bachelor's thesis here at the Helmholtz Institute at the Comparative Microbiome Department, short COMI. And I'm working with around 100 samples to research the impact of fluorphenicol, that's an antibiotic, and amamectin benzoate, that's an antiparasiticum. The impact of those two on the sea urchins, on the gut microbiome for sea urchins.
I'm Michelle Kickman, and I'm working right now on my master thesis. I'm comparing samples from Brazil, from an aquaculture, upstream and downstream from the aquaculture regarding antibiotic resistances.
So hello, my name is Manuel. I'm here a working student. I'm a master's student at the Technical University of Munich. And right now I'm doing a DNA extraction using a kit. I'm using root samples that have been taken from the field after a certain time. And in this project we are comparing different treatments. And probably these samples will be sequenced, and we will compare the microbial communities under different treatments and different time points.
So here we have a isolation plate. Inside there is a medium that is specific for some bacteria to grow. So we are under this system that is sterile. So we only want to keep here the bacteria that we are looking for. So we have it there. And I'm gonna take a little bit from here. And we will start there the colony on the plate.
Hello, my name is Nazani Nematzadeh. I'm coming from Iran, and this is about 9 years that I'm in Germany, and I started my education with the major biotechnology, and then in master I studied industrial biotechnology. Then I found this project from Professor Schlotter, which we had to work with the fermenters, as you see from here. This project is about the effect of flavor enhancers like monosodium glutamate on the structure and function of the intestinal microbiome. As you see here, we are working in vitro process, so it means that instead of using human and working on the human, we can simulate the human body using fermenters, as you see. Here we have chemostat system, means that it's included different fermenters that each of them can simulate a different part of gastrointestinal tract of the human body. The name of the system is SHINE. It's abbreviated from the simulator of the human intestinal microbiome ecosystem. And as you see, we can ferment the bacteria of the gut system and then make treatments on them as we want, because this system is automatically working and we can simulate the gut and intestinal of the human easily.
So here we are almost in the last part of the work that we do inside of our labs. Here in Helmholtz Center, we have several options for doing the sequencing of our samples. In this case, we are seeing here two machines, very different in the size and the technology that they apply. So here we have an Illumina MySec for short read sequencing, and here the small one is a nanopore. You can see the big difference in the size. So the big advantage of this one, beside the production of long read sequencing, is that you can actually carry almost wherever you want and plug it in the computer directly. But from this one, we will have to connect to a specific software and machines to get our final output. But in general, what we do is put in here in a pool many samples. So here we have different samples all together, and every one of the molecules is tagged with a small marker that will allow us to identify where this molecule came from afterwards when we analyze the data. So this is the work that I do most of the time. I'm working with bioinformatics and data analysis of the sequencing data that we get of all the process that we have been talking during the morning. From the sequencing, what we get is something like this. So we get a lot of data in form of sequencing that represent the microbiome of the communities from the samples that we got from the project. And what I want to know from them is how they are structured in terms of the community. I'm specifically looking for some special taxa, some special units that can be more relevant in the structure and the function of the community. That is what we call the keystone taxa. And I'm using a network analysis approach for this. So we get something like this. This is a network of a micro community. And I had to do several steps for double checking which is the role of all of these dots, because all of these dots represent different taxons in the community. And I really want to know which is the most important, or which ones are the most important of the whole community. So I need to do several analyses for rank the importance of them in the community. For example, here I have a network that represents the influence of each of the components of the microbial community. And then the colors represent what is the weight of the influence that this taxon do in the community. So here we have a good candidate for a keystone taxa. This yellow circle here is a taxon, a ASB, a bacteria, very likely a bacteria that is influencing a lot the structure of the microbial community. That means that it's very likely that if we remove this circle, the whole structure of the community is going to change or is going to get lost. So we are really interested in getting this information to improve all the process of the culturing and the establishment of the synthetic communities and the application of the synthetic communities as well.